All right, okay, so hello dears, welcome back. Welcome back, charot. Welcome back to our pre-recorded lecture on antigens and antibodies, no? And on our last video, atong last na talk about are the traits of your immunogens, no? So kita man gani no, mga itatag traits or mga qualities of our ideal man or woman, no? Ang immunogens pa kaha, di ba? No? Magpapatalo ba sila? Char. Okay, so start first again with size. Macromolecular size. All right. So unsay lang ganahan na size or unsa na size ang effective para mahimo siyang immunogen. All right. First is ang minimum nato is at least 10,000 Daltons. Ayan, pila? 10,000 Daltons. That's the required molecular weight for an immunogen to be recognized by your immune system. And typically, the best immunogens, the best immunogens, ang ilahang molecular weight is usually greater than 100,000 Daltons. All right? But there are still a few exceptions na there are still immunogens. They can elicit an immune response even if jute sila. Charot, even if gamay ra sila, less than 1,000 Daltons. But there are only a few exceptions. But generally, the rule of thumb good, the more DAX it is, the better. All right, so the greater the molecular weight, the more potent it becomes as an immunogen. Okay, so generally, di ba, as if makadako ang size good, mas ma-feel nimo, mas effective siya, mas maka-elicit siya og response, di ba? So if, of course, di ba, as the size grows bigger, no? Generally speaking, wala ko nag-specify og anything, but generally, di ba, if magkadako, usually mas ma-feel yun nimo, no? Mas maka-elicit siya og immune response, mas maka-elicit siya response from you, di ba? So same here pa din in immunogens, kamog yun. Immunogens atong gipang chikahan here. All right, immunogens, of course, the bigger the better, di ba? So because again, mas ma-recognize siya by the immune system, hence, mas naay immune response na ma-produce. Alright? Okay, so muna na siya ito ang rule of thumb, guys, ha? We always choose the DAX. Charot. Okay. Alright, so at pila ang minimum? 10,000 Daltons. Pero ang best, usually are greater than 100,000 Daltons. My gosh, Mark. Ang aga-aga pa. Okay, alright. Now we go now to next, its composition and complexity. Like, what substances are composing this immunogen? Kinsa tong effective? So in this list, in this order, muna siya ang effectivity of the composition in being an immunogen. All right? So first is, of course, proteins. Your proteins are the most immunogenic. Why? Because your proteins, diba, recalling your biochemistry, they have 4 structures, like primary until quaternary. And as you can see, mas maging complex siya. So mas magkadako po yung sizes, no? And mas, in a way, mas complex iyahang structure, so mas maka-elicit og immune response sa body. Alright? So proteins ang pinaka-immunogenic, okay? Or mas pinaka-maka-produce or maka-elicit og immune response. The next one is your carbohydrates. They are less immunogenic than proteins, and they commonly occur as glycoproteins or glycolipids. So when you say glycoproteins, proteins na ay attached na mga carbohydrates. And lipids na ay attached na carbohydrate portion. Okay? Alright. Next, ang pinaka-least na gyud or non-immunogenic are your lipids and nucleic acids. So non-immunogenic unless attached to a suitable carrier molecule. So example, glycolipids. So if lipids in itself, dili siya non-immunogenic, alright? So meaning dili siya ka-produce og immune response. Pag adan mo siya og, let's say, a carbohydrate, so kanang example, glycolipid, alright? So mahimo na siyang antigenic. Joke, sorry. Immunogenic. Alright? Ayan. Gets? Okay. Alright, so kabantay muna na ay ubang mga, let's say, mga prosthetic valves nato, no? Or like kanang mga foreign materials usually, or kanang gina-transplant. Usually, atong butangan og, I think one of their composition is lipids food. Yeah, because of this reason that they're not, lipids in itself are not that immunogenic or they're not immunogenic good. Alright? Okay, so that's for composition. So again, on si pinaka-immunogenic, proteins. Alright, in this order ha, proteins ang pinaka-immunogenic, followed by your carbohydrates, and then your lipids and nucleic acids. I'm sure kabalo po sa nucleic acids, di ba? DNA, RNA, mga ganun. Alright. Okay, ayan. So now we go now to foreignness. Okay, so when you say foreignness, how foreign is that immunogen from your species? Like, the farther it is from the species of you, the more immunogenic it is. Alright, ayan. So, is the degree, again, to which antigenic determinants are recognized as non-self, meaning your immune system ang good has the ability to recognize na, uy, ako ani, uy, dili ni ako Sana all, kabalo mo yun ana. Dili mga agaw. Charot lang, tsaka. Okay, alright. So, again, that's the point of foreignness. Alright, an immune system, again, can normally distinguish between the self and the non-self. So, makaingon siya na, kabalo yung immune system kung unsay naani mo gyud, like kabalo siya kung unsa ang for you and ang dili for you. Wow. All right, okay. So kabalo siya kung unsa imong antigens, kabalo siya og kinsa tong katong mga antigens na dili imuha. All right, okay. So the rule of thumb, the more distant taxonomically the source of the immunogen is from the host, the better it is as a stimulus. So example, ikita humans. Let's say you have an antigen from animals and you have an immunogen from let's say, humans, no kita. Let's say you have an immunogen from animals, you have an immunogen from plants. So, asa man ang mas immunogenic ato, ang animals or plants? Of course, if wala ko na sa iyo, it's the plants. Kaya medyo mas layo sila o taxonomy compared to us. Ang animals man good, let's say, some of the mga chimpanzees and all that, they quite have the same species sa atua. They're quite near lang sa atua. No, in terms of taxonomically speaking and sa species, if wala ko na sa'yo, oh my gosh, then of course, medyo dili kayo immunogenic ilahang antigens to us. Whereas, ang plants, nalayo gyud siya in a way, so mas maka-elicit og immune response ilahang immunogens gikan sa plants. Okay? Nagets lang? I hope wala ko na sa'yo. Alright? Okay. Ayan. And the last trait is antigen processing and presentation. Now, magtingala mo na, hi sir, na day presentation po ni mga antigens. Yes. Graded niya siya, guys. Joke. Apilin sila ang final requirements. Joke. Okay, so, of course, your antigen, for it to elicit an immune response, it should be processed. Okay? It should be presented. Okay? So, yes, mag-ano sila guys, mag-interpretative dancing. Charot. Role play. Charot. Okay. Push na, Mark. I-push rin yung joke. Okay. So, involving dapat, your antigen is digested enzymatically by the cell. Example, phagocytes, no? Your phagocytes, generally. And once they are digested, katong mukuha ra og parang a portion from the antigen and iyang i-complex na with what we call the MHC molecules and i-present na siya to responsive lymphocytes. Okay, so kailangan dyan siya ma-present or kailangan ng antigen presentable to your responsive lymphocytes. Because if not, then it would not be able to produce an immune response from the host. All right? Or the antigen can also complex with your MHC. Now, your MHC or major histocompatibility complex, this is an antigen found in your cells, okay? All right? Or parang peptide found in your cells that, again, iyahang purpose is iyahang mo-complex siya with the antigens processed by your cells, kaya iyang i-present to your T cells, okay? Para ang T cells na ang mo-signal sa other cells na naay antigen na nakita. Parang ganun, okay? So, medyo guys, nalimot ako sa process or medyo dili ko medyo malibog pug ko light. So, I'll let Ma'am Teddy explain, ha? Kanang MHC, especially ang MHC. You'll have that in your lecture. Alright? Okay, alright. Ayan. So, ang point lang yun is dapat ma-present siya. So, example, this is a phagocyte, no? Ayan. So, ihang i-digest ng antigen. So, ni-present na siya o a portion. So, of course, this is a T cell. And usually, this is a T helper. Okay? T helper cell. A type of T cell, guys. CD4. Okay? CD4. Alright. T helper cell. By the name is of helper. Alright. So, the MHC molecule is this one. So, mura siya, a peptide usually, like it's a structure na nag-protrude from the surface of the phagocyte. Alright? So once gidigest na sa phagocyte ang antigen, mukuha siya og bisag like parang a portion lang of the antigen. And iyan ang i-complex with the MHC. Alright? So this is the processed antigen. And then of course, the T cell receptor, iyan ang i-notice. Alright? And then once i-acknowledge na sa T cell, musana na siya ng mga signals sa other cells sa immune system, especially sa B cell and the other T cells. Alright? Okay. Para mo produce ng immune response. Yes, sinana siya ka-amazing, actually. And ka-complex, guys. Naglisod ako diri, Char. Okay. Alright, ayan. Okay, now we go now to next what we call your epitopes. Alright. So let's say, di ba, your immunogen, kailangan dako iyang size, no? But actually, even if kailangan o dako na size, only a small portion of the antigen ang needed para ma-recognize siya by the cells in your immune system. And that is what we call your epitope or determinant site. So this is the, parang, let's say, as a protein ba, like a structure in your antigen that can be, or that is now recognized, or that can be recognized now by your cells in the immune system. So it's a key portion of the immunogen, all right, that attaches to the antibody or your T cell receptor. Okay? All right, so epitopes are molecular shapes or configurations that are again recognized by your B or T cells. So ina na siya ka-interesting good, ina na siya ka-amazing po, no? So on the surface of your antigens, usually they have these shapes, molecular shapes or configurations that can be noticed, or that can be noticed by your B cells or T cells. All right, that's amazing. So here's an example. Ayan. So ano na siya ha, figuratively lang siya pag-drawing. All right. So these are your epitopes, of course, and you have an antibody pod na kanang specific yun to this epitope. Like naagid siya same size, same configuration. All right, na mag-react sila together. All right. Ayan. So that's in a nutshell a rough drawing of your epitopes. Okay. All right. Now, what we call, there's also another term that we we denote, or there's another term that we call, there's another term na atong i-discuss, and that is your haptens. Now, haptens, these are just some small molecules. Now, by itself, they are not immunogenic, but they are antigenic. I hope na-gets na ang difference ito. So, they are antigenic, but they are not immunogenic. But if they are complex to a larger molecule, then they now become an immunogen. All right? So again, haptens, non-immunogenic materials, then when combined, when combined with a carrier, meaning a larger molecule, it now create new antigenic determinants, meaning pwede na sila maka-elicit og immune response. Kaya na new antigenic determinants and then new binding sites. Okay? All right. But it only has one epitope. Naa sa usa ra ka binding site. Okay? All right, ayan. And once antibody production is initiated, the hapten is capable of reaction with antibody even when the hapten is not complex to a carrier molecule. So, even if, because of the carrier molecule, so what happens is the immune system recognizes now the hapten, so small molecules lang, okay? So, now that the immune system has recognized these haptens already, so if nanapoy another encounter with those haptens na wala na carrier molecule, all right? Like siya yung usa, maka-respond ng immune system. Kaya nakabalo naman siya, okay? Or na-recognize naman niya itong haptens before tungod atong sa carrier molecule, okay? Na-gets ra? So here's an example of a picture. So hapten molecules, again, mga small molecules na by itself is non-immunogenic. So let's say we have a large carrier molecule and nag-combine sila. Now this is now a complete immunogen. Alright? So, kanisya now ang ma-create og immune response. Alright? Okay. So, your hapten, it confers the antigenicity, meaning how, antigens, how foreign or iyahang pagka-foreign, and usara yung epitope, usara yung binding site. Whereas, ang carrier ang nag-produce sa immunogenicity. So, bigger molecular weight. Alright? So, example, haptens, no? So, first reaction pa lang ni na encounter sa imong body. So, since ang haptens, na-combine man siya sa carrier molecule, this will now elicit, di ba? Immunogen na siya. This will now elicit an immune response from your body. Okay? Now, let's say, second exposure, haptens na lang, kanira sila, wala na ni, of course, would nana ba immune response from the body? Yes or no? Yes. Ma-recognize siya by the immune system. Kay, na-recognize naman siya before through the help of this, combination of the hapten and the carrier molecule. Gets? Alright? Okay, so your immune system now will attack the hapten molecules. Bahalag gamay ra siya, bahalag wala siya carrier molecule. Alright? Okay. Ayan. Now we go now to the relationship of the antigens to the host. Alright? So some terms. Okay, so they can be placed now in broad categories according to their relationship to the host. So first is autoantigens. These are antigens that belong to the host. Yes, we have antigens that are ours. Okay, so usually examples sa inyong blood bank. Praise, yes. Blood bank, you will encounter different antigens. Ato ang blood groups, mag-think mo na we only have the ABO and RH blood group. Guys, you are wrong. We have already 36 ba, if I'm not mistaken? 36 ka blood groups sa ato ang blood, sa atong human body. Yes, we have 36 blood groups. And you'll discuss that in your blood bank. So, God bless, guys. Okay. So, that's what you call as autoantigens. Kita mismo natay antigens. Alright? That's not necessarily, so sir, mo ka na antigens, di ba? So antigens foreign, ganun wala ni-react. Because again, di ba, your immune system knows between the self and non-self. So kaning atong autoantigens, our immune system knows that these antigens are ours and that they do not cause infection. Unless na lang in the future or na mga cases wherein the immune system attacks our own cells because the immune system cannot distinguish between the self and non-self. And that is seen in diseases of autoimmune ang origin. That's why natay autoimmune diseases. All right? Okay, ayan. That's the point of autoantigens. For next, allo, excuse me. All right, allo antigens, by the name itself, allo, from other species. So like human to human, animal to animal. So other species. All right, and usually they can elicit an immune response. Example, if ang iyang antigen nabutang nimo, all right, so that's something that is non-self, okay? So mo elicit siya immune response. And where do we consider that? Usually in transplantations and transfusions. That's why it's important that we, di ba, in transfusion and transplantation, we test for mga antigens coming from the donor, all right? Usually para dili siya maka-elicit og immune response sa receiver. Kaya pwedeng mamatay ang receiver. Alright? Okay, that's allo antigens. And next is hetero antigens from other species. So example, humans and antigens sa animals, antigens sa plants, okay? Alright, and microorganisms. So these are hetero antigens. Okay. Alright, now we go now to your adjuvants. Okay, so what are adjuvants? Okay, alright. So these are just adjuvants. These are just substances that can increase or strengthen the immune response of your body, of a body, to an immunogen. Okay, so usually these are used in vaccination. Okay, so ang adjuvants are substances that can be added sa imuhang vaccines usually para mas mo intensify, okay, ang immune response of your body. So para mas makadaghan or mas daghang antibodies ma-produce ang imuhang adjuvant, imuhang body. All right. And we have also an example of an adjuvant is ayan, nara dere. All right, aluminum salts and your Frohn's complete adjuvant. And your complete adjuvant, it contains mineral oil, emulsifier, and killed mycobacteria. So, there are some vaccines, nalimot lang kung saan na vaccines, ang naay adjuvant na Frohn's complete adjuvant. Okay, all right. So what it does is, of course, again, ang point lang yun is para mas mo elicit og stronger, all right, and intensify ang immune response. All right. A large number, yes, it can attract a large number of immune cells at the extraction site. So by doing that, of course, mas daghang cells involved, mas daghang antibodies na pwedeng ma-produce, and mas daghang cells ang maka-remember, di ba, memory, all right? And mas daghang cells pud ang mo react. Okay? So in a way, parang mas makirig or dili, mas hyper, all right? Or ma-hype imuhang immune system in a way. All right? So usually seen in vaccines. Okay? All right. So some aluminum salts and Frohn's complete adjuvant. But I think if I'm not wrong, kani siyang Frohn's complete adjuvant, I think wala na kay siya gigamit kay naa siya mga major side effects. I think if wala ko na sa pa to some patients. All right? Or some na magpa-vaccine. So munang wala na kayo siya gi-use. Okay? If I'm not mistaken. All right. So that's the purpose of an adjuvant. Okay. Again, ang point lang yun is it is used for adjuvants. It's for to make sure that the immune response again mo generate a greater, okay, or intensify niya ang immune response. Again, why? Para again mas daghang cells involved, mas daghang antibodies na ma-produce, and eventually mas stronger ang immune response of the body to that particular let's say antigen ba or substance na gi-introduce sa vaccination. Alright? Diba? That's the ultimate goal of vaccination. We want to produce antibodies. We want to produce, generally, antibodies good to combat the disease na ato ang gi-vaccinan. Okay? Alright, ayan. Now for the... Kaya pa ba? All right. Okay, sige. Let's push through. Okay, so now that we're done with antigens, no, so we now discuss its counterpart, of course, yung partner. And that is your antibodies. So these antibodies, again, muna ang respond usually sa imuhang body against the antigen. All right? So again, kinsa mo produce sa imuhang antibodies, your B cells. Okay? Pero ang pinaka-specific yun, guys, are the plasma cells. Okay? Plasma cells. So plasma cells are a type of B cells na ni differentiate. So ang B cells mo differentiate na siya to plasma cells. Okay? All right. In the presence of an antigen. All right? Yes, ayan. And the plasma cells na gyud ang mo produce og antibodies. Di ba? Recall in our CM sa ato ang class sa AUBF, di ba? Unsa tong disease na proliferation of the plasma cells, your multiple myeloma. All right. Okay, di ba? So again, plasma cells, they now produce your antibodies. Okay. So what are antibodies? Your antibodies are immunoglobulins, IgS. They are, again, specific portions of the liquid portion of the blood seen in plasma ba or serum and in many body fluids. Saliva, tears, colostrum. Colostrum, guys, is colostrum, again, is the first milk of a mother, diba? Of a lactating mother. Of a lactating mother. So, kana magpatutoy, yes, mga nagpapatutoy dyan, pero wala pang baby, charot. Bastos, okay. Alright, basta milk of a lactating mother, the first milk, colostrum. Alright, so it contains antibodies. Alright, ayan. Okay, and when subjected to electrophoresis at pH 8.6, they usually go to the gamma portion of the electrophoresis or in the gamma band. That is why they were known originally as gamma globulins, all right? But throughout the years of studying, na may other antibodies na dili lang sa gamma band moadto pag electrophorese. That is why gi-change to immunoglobulins, okay? But generally, ang first name is gamma globulins. Because again, why? They first pag-study, they all go to the gamma band of your electrophoresis. Here, ayan. So as you can see, ayan. So that's electrophoresis. This is the albumin portion, alpha 1, alpha 2, the beta globulin portion, and the gamma. So most of the antibodies daw mo reside siya here sa gamma band. That is why gitawag silang gamma globulins. But then, throughout the years ng pag-study, it was discovered na not all antibodies have gamma electrophoretic mobility, meaning dili sila tanan mo adto diri. Alright? That is why gi-change ang ngalan. Alright? Okay. Ayan. Okay, alright. So, yeah. Okay. Alright, so for the next video, we'll now discuss further the antibodies. Alright? And we'll start first with its structure, and then finally its types, and then the end. Okay? Alright. Ayan.